Okay, well, welcome everybody. We're excited to find out what the teams have done and what you've learned this summer, and we're looking forward to hearing from you today. I'm Mitch Aiken. I head up outreach at Caltech in the Center for Teaching, Learning, and Outreach, and the Summer Research Connection is one of our big flagship projects for the year. So we've had a very busy summer. We had 51 students participating this summer. We had about 20 groups, so there were lots of activity, and all remote, unfortunately, again this year, but we hope to have it all back on campus next year. So with that, I'll just pass it around and we'll do introductions. Charlie, would you like to say hello?
Yeah, hi, my name is Charlie. I'm an incoming senior in high school. I think I might be the only person that's not from California. Correct me if I'm wrong. I live in Weehawken, New Jersey, which is about 15 minutes from Manhattan.
Welcome. Okay, Jeff, I'm just going to go on the line on my screen. Jeff?
Hi, I'm Jeff. I'm a current student at Caltech, and I work in Paul Bellan's lab, and I worked with Omar on his project this summer.
Thank you. Kevin?
I'm Inez, or Kevin Weiss. Inez is my daughter. I am a 17-year teacher, secondary science and robotics and like STEM competitions and the like. Happy to be here.
Thank you. Paul?
I'm Paul Bellan. I'm a professor at Caltech in applied physics, and I worked with Jeff and Mitch on setting up a summer project that you'll hear about.
Okay, Omar.
Hello, everyone. I'm going to be a senior at Santa Ana High School in Santa Ana, California. Happy to be here.
And Elliot?
Elliot Meyerowtiz. I'm a professor at Caltech in biology and biological engineering. And I've worked this summer with Eldad, who has supervised Joshua.
Thank you. And Matthew?
Hi, I'm Matthew Graham. I'm a professor at Caltech in astronomy. And I've been working with Charlie and Hexon this summer. And independently, my daughter has been working on another program as part of this thing. Nothing to do with me, all through her school. So.
Yeah. Okay, Eldad.
Hi, my name is Eldad. I'm a postdoc at the Meyerowtiz lab. I work on an algae, and Josh has been working with me on a related project.
Thank you. Josh?
Hi, everyone. I'm Josh. I'm an upcoming senior at Granada Hills High School in California, and I'm working with Eldad in Professor Meyerowitz's lab.
Thank you. And Hexon?
Hi, my name's Hexon. I'm an incoming senior at Santa Ana High School, California.
Thank you. Okay, nice to meet everybody. And we'll go ahead and get started. Did you guys decide which group would go first, or we'll make a decision on the spot? Any volunteers? All right, Josh.
Okay, let me share my screen. Does this look good?
Yes.
Okay, great. Hi, everyone. My name is Josh, and this summer I've been working in the Meyerowitz lab under the supervision of my mentor, Eldad. Today, I'm going to share a bit about the field of color per regeneration, what I've been working on, and where we hope to go in the future. So first of all, our plant of choice is the Caulerpa brachypus, which is a small to medium-sized seaweed, which is notable because it's all one single cell. As a plant lab, we have a twofold focus and interest in this plant. First of all, because plants make up the majority of our food, medicine, and fabrics, any advancement that we make will have a lot of real world impact. Secondly, because it's such an extreme example of a single cell, hopefully any findings we make in this notable example will be able to carry over to more normal cells like the ones we would find in our body. In the right, you can see the photos of what we use in the lab. So we start with a very small cutting, and then within about 3 weeks, we have the cuttings have grown, and they've also developed blades and offshoots. Previous findings in the lab before I got here were that the greenness of the caulerpa will follow a circadian period based on illumination. The lab ran an experiment with 12 hours of dark followed by 12 hours of light for about 2 weeks on the plant. So here on the left, we have the dark image, and on the right, we have the light image. The right looks a little bit odd because of the way the camera and the projector works, capturing just a snapshot of the light. But to the human eye and to the plants, it just looks like a bright daytime setting. So on the left, we can see that there's a big difference between the stem of the blade and the edge. Here it's much darker and notable, whereas down here, we can barely even see it. Similarly here, the base of the blade is a bit darker than the top. Whereas during the light period, we can see that this whole blade is about the same color. Similarly, this whole blade is about the same color. So in this video, we can see that as the plant grows, the greenness goes in and out based on the day and nighttime. During the day, it'll become more green, whereas during night, it'll recede, which is also shown in this graph. We predict that this is due to chloroplasts moving around inside the plant, perhaps in order to cover more surface area during the day to help with photosynthesis. One thing to notice from this graph is that the greenness starts to come in before the night period comes up, and also the greenness comes out before the day period comes up, showing that the plant is anticipating this circadian rhythm. After 2 weeks of 12-hour day, 12-hour night, and so on, we instead subjected the plant to 24 hours of constant daytime. And what we saw is that the plant continues to anticipate this 12-hour cycle, even though it's not receiving any of that lighting. So now for my part, we're trying to follow up on this project by using differential illumination. So I wrote some Python code to work with the BeagleBone Black, which is a little computer similar to a Raspberry Pi, and also with a DLP, which is a projector, in order to project these checkerboards over the plants. Before we had a 4x2 well, and so this checkerboard is 8x2, so that every plant will have one dark period and one light period in order to break it up and see how those anticipatory patterns react in a local versus central way, given that it's all one single cell. In order to capture the images that we looked at, now that instead of a homogeneous dark versus homogeneous light, now that it's split up into dark, light, we need to manually set the Raspberry Pi camera that we're working with in order to capture one dark mode picture and one light mode picture almost simultaneously in order to get every single part of the plant in good focus with the camera. So I'm working on that code right now, whereas I had already completed the checkerboard. In the future, we want to look at first projecting light dark directly on the samples. So that would be, if we have a blade here, it would be projecting just around the outside so that we can be more specific with our research. And we can also test how good our experimental setup is, how much the light diffraction diffuses. Secondly, changing the camera and the projector setup, which is whether we have the camera pointing down and the projector coming in from an angle, or we have the camera projector both from an angle, or we have the camera on top and the projector on the bottom of the wells. And lastly, spectral characterization of the LED versus DLP wavelength and intensities. The checkerboard that I wrote are for the DLP, which is a projector similar to maybe a movie projector, but other experiments that we used had an LED, had LED lights, which is just a bunch of little LEDs around. So we want to test the wavelength and the intensities of the LED and the DLP to see whether that will have an impact in the differences that we see in the experiments, which could be in regards to the growth of the plant, the changing greenness of the plant, and other factors. Finally, thank you to Professor Meyerowtiz for taking me into his lab, Eldad for mentoring me and communicating with me on a daily basis, and as well as everyone else who has helped me over the summer and whose work I've built on. Does anybody have any questions, feedback, or comments?
I have something. I assume it's very rare that plants would lose their green color at night. Are there any other plants or single cell plants that do this that you know of?
Well, not that I know, but one thing to think of is that this is a seaweed. So first of all, it has a very light color. It's not like a tree outside where it has an intense kind of green, vibrant color. And it's also used to being way underneath the water, where it's not necessarily getting the strongest sunlight in the first place.
Makes sense.
So because of COVID, I became amateur gardener and learned more about plants than I knew before, which was next to nothing. I got some grow lights in my garage, and I looked into that, and I discovered that there were grow lights that purposely, I think, didn't have green. And I understood that it's because the plants absorb red and blue, and they reflect green. So that's why. So the plants actually don't use the green; they use the other colors. So do you have any thoughts on that, and am I correct in what I'm saying? That's
Yeah.
fine.
So the green color that we see is because the green kind of reflects off, whereas the other colors are absorbed. One interesting thing that came up when I was talking with Eldad is there's all sorts of possibilities for different colors of lights, different intensities. And so if I remember correctly, there was an experiment that showed that if you flash a blue light in the middle of the night for the plants, if you just flash a blue a couple of times, then it'll completely reset the cycle of the plants, or it'll kind of wake them up.
Oh.
Yeah, so color can do all sorts of things for plants.
People are sensitive to color too. I mean, in northern regions, people get depressed because they don't have enough sunlight, and people have lamps to shine on them, and they seem to not get depressed because of that.
Josh, if you want to end the screen sharing, then we'll have you on a full frame.
Yeah, there we go.
Okay, there we go. Okay, perfect. Any other questions from anybody else? Any of the students or other mentors?
I don't really have I don't know if it's necessarily just for Josh. So the chloroplasts removing is kind of the reason that you're saying that the color changed. What would be the advantage to moving your chloroplasts for the evening, kind of packing up and stuffing them away and then bringing them back out when the sun comes up? Does anybody have an idea on that?
So I'm sure the other people in my lab can answer better than me, but from what I know, what we're thinking is that maybe it's not exactly to fulfill a purpose like you or I would think, oh, it'll help you with energy or whatever, but more that as the plant is kind of regenerating and the cytoplasm is moving around, maybe the chloroplast just happened to move in kind of in the natural flow of the plant.
Yeah, my answer to that is we don't know. You can think of all sorts of plausible reasons for this, having to do with light capture or the chloroplast getting down to out of the light when it's too bright, or the spectrum changes, or what have you, but we just have no idea.
If I may add my own perspective, I would rephrase the question: what are the dynamics that drive this purpose? What underlying mechanism drive this behavior? As if like when you have a pendulum and it goes down but then keeps on going, so you know there's an interplay between gravity and inertia, so that thing drives the thing over and over. And specifically with biology, we could try to ask whether this adds some stability to the system in the sense that it survives, and we keep seeing it as alive and not just disintegrates into something dead. So it's just a rephrasing of the question, but I think it puts a focus on maybe something that we could actually track in the lab.
Yeah, there's some things that'd be hard to track too, though. An example is that chloroplasts exchange that they have DNA, they have their own genomes, and they exchange their genetic material as a way of repairing DNA damage, which would occur from light photo damage. And so possibly they all go together, so they're close together and can trade their genomes. Who knows? You can think of 100 reasons There are almost no experiments that would help you with any of them. And that's pretty much how evolutionary biology is all over. There's always some Rudyard Kipling sort of story about how the giraffe got its neck, and there's never any evidence.
What retains memory of the diurnal cycle?
We're not there yet. In many different organisms, there has been identified an oscillator, which is a genetic network.
Okay.
Some others, it's not a genetic network. I cannot say what is the case here. One thing that is interesting from taking on what Elliot said is that often people expect that when you find a mechanism, it's conserved across evolution. From the little I know, I'm not coming from molecular biology background, from the little I know in circadian rhythms, even the genetic network is not conserved, not even in its topology. So the ingredient, it's not only that the ingredients are different, but it's the number of ingredients and so on, the components. There is evidence. Well, okay, I'll stop here, but I'm
Yeah, that's a discussion we can continue a lot.
Yeah, I'm happy to keep on discussing this as well.
Thank you. Thanks, Josh. Very good. Any other questions for Josh? All right. Who would like to go next? All right, Omar. Take it away.
You guys see that? Yeah.
Yep, we can see it now.
That's good. All right. So, hello everyone. My name is Omar, and today I'm going to be presenting about constructing and using a gradient condenser from atmospheric ion measurement. And this project was supervised by Professor Bellan and Jeff and Kevin. Thank you guys. Caltech. So as for what exactly I'm going to be going over with you guys is the context, construction rundown, theoretical technicalities, data analysis, conclusion, and some references. So as for the context, the whole focus of this project is ions. And to put it simply, ions are charged particles in the air, which always happen to be present. And in our project, we went over some research papers, and one research paper, to be specific, was by Professor Flagan at Caltech. And basically, it was just a whole collection of reviewed ion condensers over the past century. But this project specifically, we focused on Gerdian's ion condenser, which was developed in 1905. And there's actually a diagram on the right of Gerdian's condenser. And to basically, put it simply, air ions go through the cylindrical capacitor through a fan, and the outer electrode repels the air ions to the inner electrode, and doing so actually creates a small current. And with that small current, we were actually able to amplify this, immeasurable current into something that we actually could measure with the voltmeter. So here's the actual device we created over the summer. So on the left is the materials, and on the right are some pictures. So on the top left is actually the green condenser at the site we actually decided to go to and use to gather some data. On top is the of the circuit, and on the bottom itself that we use to amplify the small current. And the bottom right is it's actually not my picture, but I just didn't want to deassemble the usable project yet. So the formula that actually ran this whole project is to your right. So what each variable means is that the C is equal to the air ion concentration, the I is equal to the current from the ions, E equals the charge of the electrons, and omega is equal to the volumetric airflow rate. And there's actually an equation within an equation where omega is equal to the surface area of the bigger electrode on the outside, subtracted to the smaller electrode on the inside, multiplied by the rate of change, the velocity. So here's the actual circuit. No, it's a lot, but to put it simply, small current, we were able to through a small correction with the potentiometer, and also through thanks to Ohm's law, we were able to take this small current and actually convert it into something that's actually measurable, a voltage. And then through that, with an op amp, we amplified, what was it, 10 to the negative 12, a picoamp, to around a 10 to the 1. So we were able to actually measure the current with the voltmeter. So here's actually Eden Canyon, or the waterfall we actually went to gather some data, which happened to be in Pasadena, California. So right behind our backyard. And here's the exciting part, the data we actually collected. So we decided to collect data from three different spots from the waterfall. So here's the set of data from the farthest away we decided to take from the waterfall, which happens to be around 85 meters away. And we decided to organize the data through a histogram and a scatter plot. So, and we also gathered the measured mean air ion concentration, which happened to be for this set of data 826, and standard deviation 286, and then both in ions per cubic centimeters. And also, we also took data every 5 seconds. So, and here's another set of data that we took from 25 meters away from the waterfall this time. Again, histogram scatter plot, measured air ion concentration mean 1455, standard deviation 402. And the set of data we took was around 5 meters away from the waterfall, which is the closest we got to it. And again, histogram scatter plot, measured air ion concentration mean happened to be 1857 this time, and the standard deviation happened to be 494. And if you took notice, the standard deviation actually went up the closer we got to the waterfall. And what's special about this, the last data, is that the fan wasn't actually running, or it was running, but just not as fast as it's supposed to the first minute. So if you can notice in the scatter plot, really young, there's like some low points. And so what we did exactly is just straight up got rid of the set of data, just have a more presentable set of data for you guys. Organize magnitude of the data and scatter plot with the three set of points, three sets of data, and we got this relationship where we can't really conclude, because after all, it was only three set of points, so we can't say that it's decaying exponentially, or it's growing, or it's parabolic. We just don't know exactly. As for the conclusion we came up with, is that there was actually another group of scientists that actually gathered data at a different waterfall using a gradient condenser. And what we can conclude is that their set of data is not exact, but it's strongly similar to ours, where the closer you got to the waterfall, the more ions you were able to measure, and the farther away you got from the waterfall, the less ions you were able to measure. This is actually a picture of the waterfall that group of scientists actually went to and gathered data. Completely different location in Austria, and much bigger than ours. As for some limitations that came up with, that we were only able to gather one set of data, and with that one set of data, we were only able to compare it with that of another box. And also, we ran the instrument without complete noise calibration, so a couple of effects came into play due to the change in weather and some other noise. That's what we can use with this instrument, is that we can potentially use the instrument to measure some power lines and voltage boxes locally, to technicians, or also possibly to submit the product to a local science fair. I just want to emphasize again, thank you so much for Caltech giving me this opportunity to be able to research under Professor Bellan, and I had a good time over the summer. Thank you.
Okay.
Thank you. So there was some engineering involved in this one, in actually building the condenser, and then actually some field research. So that had a nice combination of several different things happening this summer for your project.
Open to any
Anything
questions.
Yeah.
Yeah, I have a very naive one. Why would the ions be different whether you were close or far from a waterfall? How does a waterfall make ions?
If I'm not mistaken, the water itself, water is falling from however high. So the water is falling and it breaks through the air particles and thus create ions, if I'm not mistaken. I think that's how it works, or how it actually goes down. Joff, if you want to correct me, or Kevin.
You got it pretty much right. I mean, a waterfall is a very, very violent process full of lots of tumbling, water molecules, some of which are ionized, falling off into the air. I think it's almost physical how the ion concentration increases theoretically.
Yeah.
So would it correlate with humidity then?
Yes.
Yeah, I don't know the answer myself. My suspicion is it has to do with the rubbing against the rocks, like walking on a carpet. That you can make, you can charge things when you rub things together, but I'm not sure if that's the only explanation.
Eldad, did you have a question or comment?
It was more of a comment or suggested. We were traveling in Oregon next to Zigzag Falls, and they were making a case over there that near waterfalls people feel happier because of those free agents coming from the ions. And so, yeah, exactly. You could try to then correlate the mood of people as a function of the distance from the waterfall and see if you find a correlation.
I think there have been many devices created to change the ion content in our air to supposedly improve our health and our disposition. So either there's something to that, or there's a lot of people that buy into it, because it's a whole industry, I think. So I think more to the point for this project is this is all part of looking at indicators for the quality of air in general. Can anyone speak to that? It's like, why do we want to know what the ions are doing and what the concentration is?
Jeff, do you want me to answer this one?
Yeah, sure. Go for it.
Okay. This is my thought process. So, plasma is made up of free electrons, right, and positive ions. And with this device, we can. Oh, okay. Oh, sorry. I thought I was having the. You guys can hear me clear, right? Because I'm getting this message where it says internet stable. Can you guys
Pretty
hear me
well.
good?
We can hear you. Your picture is freezing, but we can hear you okay.
Okay, cool. Doesn't matter. Yeah, my face doesn't matter. Okay, so with this, we're not going to use this exact device, this homemade device to measure plasma and atmosphere, but we can use a more I guess expensive, more realistic device to actually measure some atmospheric plasma with this gradient inspired condenser. That's what I'm getting out of it.
Thank you. Any other questions or comments? Eldad?
I actually have a question. It's following up on what Professor Bellan said. How would you test whether it's more effective, say, surface tension or rubbing against rocks, like what they extract from the rocks, or whether just water by itself? Is there other standard ways to test that in the lab? The generation of ions, sorry. The question is about the process, the essentials of generation of ions. I for some reason assumed that it's the water droplets stirring and not the process of rubbing against another material.
Well, I don't have an answer for that. So, yeah, I should say that I do plasma physics, and plasmas involve working with vacuum chambers and often high power and high voltages, and are dangerous. And the interesting thing about this project is that it's not the kind of plasma physics I'm familiar with, but it's nice that it doesn't involve a vacuum chamber and doesn't involve anything dangerous. So it's a very good introduction. And the method is actually very similar to a probe that is commonly used in plasmas, where you put a wire in and you measure the currents to the wire, and then you can deduce the density of the plasma and sometimes the temperature of the plasma. So it's kind of a close cousin to what I work on, but I don't actually work on this. However, the history of it's interesting. If you look up the history, it goes back to the days of Benjamin Franklin and Alexander Volta. People in those days were studying atmospheric electricity. And then actually, when I was around 20, I had a summer internship as a university student for a guy who was measuring atmospheric electricity. And I'd forgotten about it, and it occurred to me that I've almost come full circle, going back to what I was doing when I was about this age.
Nice.
So I think it would be interesting. You know, when you're at the seaside, you always get that smell of ozone as well, which I guess must be a similar ionization that's going on with the water. It would be interesting to see whether you measured the same degree of the effect just by a rough sea as you do by the waterfall, because that might give you some commonality between the physical processes that are going on underneath. Is it just the turbulence and the pounding on a hard surface? Or is the, you know, are you getting some particular turbulence effects because of the gravity fall with the waterfall that's increasing surface area to atmospheric exposure or something like that, which might be generating a stronger ionizing mechanism?
Sounds like a good excuse to go to the ocean.
Yeah. Next year, the experiments at Malibu for 6 weeks.
I'm heading to Santa Monica Beach for scientific purposes.
Yeah, just hang out by the pier. Yeah.
Sounds good. Okay, well, thank you, Omar. Very good. Thanks for all your hard work. And now we will turn it over to the Graham group. Who wants to kick us off?
Well, I just have to say that one of my group members' power just went out. He just emailed me, so.
Oh, Hexon has lost. Oh no.
Oh no.
What do you want to?
Yeah, because the first half of the presentation's his, so.
Oh no.
Oh boy, timing is everything. Oh gosh.
And you're muted, Mitch.
Yeah, sorry. So more than just his internet connection, his power actually went out. So that might be a challenge.
So I can. Where do you kick in, Charlie?
I kick in with results. He does the entire like background, cause, like intro to what like NumPy and Seaborn and Pandas is, and then I start with results.
All right. Okay. I mean, I can provide a quick overview so that you could then provide your results, and hopefully that gives Hexon time to reestablish his connection and then present a much better intro than me. Does that sound okay, Mitch? Yeah, okay. So the presentation that Charlie and Hexon are going to be doing together, which I'm filling in the bits for, is, so this is in the field of time domain astronomy. And Caltech is leading this with a survey called the Zwicky Transient Facility, which I'm the project scientist for, which is run down at Palomar Observatory, where we're observing the visible night sky from Palomar Observatory. We cover the whole night sky about once every 2 nights. And what this does is, and we've been running for about 3 years now. So what this does is it gives us time series for about 2 billion astronomical sources. And so there's a huge effort underway to exploit this. I mean, there's all sorts of phenomenology in there, from exploding stars to asteroids within the orbit of Venus to gamma ray bursts halfway across the universe, whatever. So for the astronomical community, this is a great data set. Now, one of the things that I'm particularly interested in are eclipsing binary stars. So most stars in the Milky Way are actually in binary or multiple star systems, unlike the sun. And what you can get is you can get a physical alignment visible from the Earth where one of the binaries will pass in front. Okay, great, Hexon's back. So I don't need to go into this and Hexon can take over.
Can you guys hear me?
Yes, we can.
Yeah, I'm so sorry. My power suddenly went out.
Well, that's fine. So I was just giving a brief intro, but please, if you want to take over, go ahead.
Yeah. Okay. Can you guys see my screen?
Yes.
Okay. Yeah, so this presentation is basically on the O'Connell effect in eclipsing binary stars. And this presentation was made by me and Charlie. And just as an introduction, me and Charlie are both incoming seniors. And she's from Weehawken High School in Weehawken, New Jersey. And I'm from Santa Ana High School in Santa Ana, California. So just some background. Eclipsing binary stars are basically two stars that orbit around each other. And occasionally their orbits will become aligned. And this creates an eclipse. Whenever eclipsing binary stars are observed, astronomers look at the data that shows how bright that object is over a period of time. This is referred to as a light curve. Individual data from the light curve is then placed on top of each other. This process is known as phase folding. And after light curve data is plotted and phase folded, it's expected that the light over a period of time coming from the binary stars should be consistent and have a sort of symmetry. However, there can be a change in the brightness that the stars give off. This effect is referred to as the O'Connell effect, and it can either be barely noticeable or highly visible. And on the top right, there's a picture of eclipsing binary stars, just an example. And on the bottom right, there's actually an example of the O'Connell effect. The peak on the right, the brightness, as you guys can see, it's lower than the peak of brightness on the left. And yeah, that's the O'Connell effect. So what's a possible cause for the O'Connell effect? The exact cause of the O'Connell effect in eclipsing binary stars is still unknown. However, astronomers have come up with several theories that may explain what causes it. And one of the most popular theories that explains why the O'Connell effect may happen is star spots. Star spots are spots that form on a star, and they're dark and have a low temperature. When star spots are distributed throughout a star, like without a specific order, they can account for the changes in a star's brightness, like for the change in the brightness. Star spots alongside gas stream impacts are also a part of spot modeling, which, when applied, has successfully explained the O'Connell effect in some cases. And even though star spots have been able to explain some instances of the O'Connell effect, there are spot models that assume that the star that is being observed has like just one giant star spot, which is highly unlikely. So while star spot theory can be applied in some cases to explain the O'Connell effect when it's done right, it still can be applied to all instances of the O'Connell effect. And on the right side, there's two pictures of sunspots, and sunspots are very similar to star spots. So me and Charlie actually used Python to kind of see the O'Connell effect, and we plotted data that we were given, and we used different Python modules like pandas, seaborn, and numpy to make the process of us making figures from the data we were given a lot easier. And NumPy in particular was really useful because it allowed us to give the plot we were making like specific characteristics, like having an inverted y-axis so that the lower brightness was lower on the y-axis, or like having a higher brightness be higher on the y-axis. And the data me and Charlie were given about different eclipse and binary stars came in 3 different filters. So green, red, and purple. And we made individual figures for each filter that contained the data for that filter. And after a figure for each of the filters was made, we made a final figure that contained all the data from each of the filters. And that figure that was made was phase folded, and it became a phase folded light curve. And this allowed us to basically visualize the data that we were given and see what it actually looked like. And on the top right, I put a picture of some of the figures we made for the individual filters. And on the bottom right, the end result of phase folding the data. And Charlie, if you want to take it over.
Yeah, so after repeating that code for all 83 data sets that we were given, we got a series of phase folded light curves for each of them. So here I've included some images of those light curves. And as you can see, these two here look very different. There was a good range of some more linear, sharp looking curves, which kind of sounds counterintuitive, but the one on the left is very straight and sharper than the other one. Rounded and asymmetrical curves. And Hexon, if you can move to the next slide. Yeah, some more uneven ones and some more dispersed ones. But regardless, all 83 of the ones that we ended up looking at, all 83 that we ended up looking at were O'Connell's. Could you go to the next slide? Thanks. So later on, Dr. Graham offered to plot our findings on the Hertzsprung-Russell diagram, which essentially shows the absolute magnitude of stars and plots them against their spectral types, which are determined by temperature. And by doing this, we can see if there are any similarities between the stars with any of these different factors. So on the right is the final image that Dr. Graham sent us. And there are 2 arrows that are pointing towards dots on the diagram. I know they're a bit difficult to see on Zoom, so I just wanted to quickly point that out. And the first thing that we saw is that the majority of them are K-type stars that live on or near the main sequence, which is that purplish, greenish chunk in the middle. And that is a great pattern, but it's not that incredibly interesting since that's where stars tend to live for most of their lives. We also noticed that there was one outlier, though, closer to the upper right-hand corner, if you can see it, of the graph, upper left-hand, sorry, with an A spectral type. Hexon, can you move to the next slide? So challenges. So even though we had these great results in the short summer, both Hexon and I had a few challenges along the way. Most notably, I had issues with learning how to use Python since I had only done very minimal coding before the summer. And it was mainly in JavaScript and some CSS, some HTML, so more of like a website background. But by the end, I do feel a lot more comfortable with the language. Obviously not a pro whatsoever, but there's been a lot of improvement since June, trust me. And Hexon's challenge has more to do with the actual coding platform itself, since he came in with a background in Python. Sometimes, especially with such large amounts of data like we were working with, Visual Studio Code, which was the platform we were using, just doesn't like to run, and it doesn't run properly. So that's where most of his issues came in, but it ended up pulling through, and Visual Studio Code did cooperate with us by the end of it. Could you move to next slide? Yeah, so as for future research, we would have loved to have spent more time doing a really in-depth analysis of the HR diagram and different locations on the diagram. So doing this may have helped us find more similarities that we weren't looking for, and just given us more time to look at that one outlier that I'd mentioned, that one outlier that wasn't on the main sequence that had a different spectral type. And our last slide, Hexon. Yeah, so that is the end of our presentation. Now, before we open up to questions, both Hexon and I wanted to thank everyone who made this summer possible for the both of us. We had a great opportunity and we learned so much this summer. So in particular, we wanted to shout out Dr. Graham for guiding us along the way, answering all our late night emails all summer, us trying to figure out how to use Python. So thank you for that. And we also wanted to thank Mitch for always keeping us in the loop with different events and webinars. Joshua Gagne, who is Hexon's teacher, who helped us both with Python, Jan Lacoste for running that college admissions event, and anyone else behind the scenes that made this program possible, that contributed to the amazing summer we had, but we might not know of. So yeah, no, thank you everyone, and open to questions.
Can I ask quick about the data that you guys got? When you mentioned that you had like a red, a green, a purple filter kind of quickly. Did that correspond to something physically? Were those like the wavelengths of light you let through?
Do you want to answer this, Charlie?
I was going to ask if you would know how to answer this, because I'm not entirely sure.
Yeah, so the red, the green, and the purple filters were just different filters on the telescope. Like, the only one I can remember is infrared. Dr. Graham, if you wanna.
Yeah, no, you're right. They're pieces of glass which are, allow certain bits of the observed EM spectrum through. So it's very common in astronomical observations to put it's no different from a filter you put on your camera, whether it's a polarizing filter or an infrared filter or whatever. But we use them because astronomical objects are black bodies. Let's assume they're black bodies, but they're all of different temperature and of different distances. And so if we can measure how bright they are in G, the green filter and the red filter and the infrared filter, we can use that as a proxy for estimating what their black body curve might be and what their overall temperature might be, or something similar like that. Or estimating, we may know how bright, we may know what their temperature is from something else. And if we then measure what we call the color is, then that'll tell us maybe how obscured the star is. If it's very red, there may be dust along our long line of sight, which the colors would show us, or something like that, if we know what the object is and what its spectral energy distribution should be.
Okay, fascinating. So it's all physical filters on the telescope.
Yeah. So okay. We have a Hexon and Charlie have seen it, but we actually have a robotic arm on the telescope which changes the filters during the night when we're observing.
Interesting.
And we always thought that would be the problem of all the mechanical failure, and it turns out, no, it's not. It's just our mechanical shutter that's the problem. That's another story.
Okay.
I have a one about the star spots. So you have these two stars and they're circling around each other. I suppose they're locked together so they're not independently rotating while they're circling around. So wouldn't the same face of the star come across every time and the star spot would be in the same place? Maybe they're not locked together like the moon and the earth.
They may not be tidally locked, but what you're seeing with the phase folded that's the repeating waveform because of the periodicity of the objects. And so the thing about the O'Connell effect is that if they're doing that and we're seeing one pass in front of the other, when they're both side by side, you would expect the brightness to be the same. But the fact that we see them different means that in the A-B orientation.
Oh, I see that.
And then the B-A orientation, we're seeing different sides of the stars.
I get it. I had the phase wrong. I thought the brightness was different when one was in front of the other.
Well,
which it would be, because the two things wouldn't have the same brightness, right?
No. So when one's in front of the other, that gives you the dip in the eclipse.
Right.
And actually from the depth of the dip and the width of it, we can estimate stellar properties like what the masses and the radius of the stars are. and that, you know, that helps with other things as well. But yeah, it's the fact that the brightness is not the same when they should be, and that's interesting. And it could be stellar star spots, as Hexon said, and that works for some, it doesn't work for others. It could also be that there's actually an accretion flow from one of the objects onto the other, and what we're seeing is when it's AB, we're seeing the accretion hotspots, and when it's BA, that's on the other side, on the back side of the star, and we don't see it. But, you know, who knows what it could be.
But it's definitely not due to there being 3 stars circling around.
I'm not certain if anyone has looked for the O'Connell effect in tertiary systems.
Might limit your hypotheses.
Yeah, it could do. I mean, yes, it's a rare enough effect just in binary stars. That's one thing.
What if the stars are not spherically symmetric? I mean, they are bright because of a fusion reaction inside, and suppose the fusion fuel is a little burned out on one side compared to the other, so it
could be
hotter on one side than the other.
So there is a thing called ellipsoidal variability, where the star is actually more of a teardrop shape because it's actually accreting material onto the other star. And so that gives you, again, in the actual shape of the eclipse and maybe the shape of the peak, it may be that you're seeing much more of a distorted effect in some than on others. So that one outlier point that Charlie indicated with the A star binary, it may be that is, you know, there's some stronger distortion going on there, some tidal deformation or something like that, which would give you an uneven effect. But I think it depends on what type of star it is about irregular nuclear burning. It would also depend on what the rotation period of the individual stars are. Again, we have stars which are rapidly rotating variables, and it would be interesting to look at those in binary systems and see whether we see something like this as an indicator of whether it's whether, you know, it's a rotational effect or whether it's a pulsational effect or something else.
Thank you all. Charlie and Hexon, as the two student team on this call, how did you collaborate over Zoom and over, well, a 3-hour time difference, I guess, East Coast to West Coast? Did you meet and work together at the same time, or just come together through email exchanges back and forth, or how did that work?
No, it was a good bit of both. We did have a super long email chain going. So at this point, I might have like 75 emails of me and Hexon just going back and forth. But we also scheduled a lot of individual Zoom meets to figure things out. I remember at the very beginning, maybe like first week of July, we were on a Zoom call with one of his teachers for like 3 or 4 hours trying to figure out some like Python issue. And I think being in a 2-person team is also really helpful for this project because of how many like different technical errors and technical issues we had. Like one day Hexon's Visual Studio Code wasn't working. So then I was the person to go do some of the phase folding, get some of the phase folded diagrams. And then I didn't mention this, but eventually my Visual Studio Code went the same way that Hexon's did. And then I was able to rely on him because we had like similar information. We were both able to rely on each other. And I think that being in that 2-person team was really helpful in this project, at least. We were both able to rely on each other and work off each other.
Good to hear. Thank you. Okay, well, congratulations to you all. Hexon, go ahead.
Oh, no, I was just going to say that I completely agree with Charlie.
Excellent. Any other questions for any of the presenters today? Okay, I think that wraps us up then. Thank you all for participating. Thanks to the faculty members and mentors and teachers who helped pull all the projects together. And we're very grateful that you are all able to be part of this summer. So thank you all, and have a good short rest of the summer. Some shorter than others now, it looks like.
Well, thank you, Mitch, for organizing this.
Yeah, thank you.
Thanks very
Mitch.
much, Mitch.
My pleasure. This was fun. Thank you. Bye, everybody.
Bye.
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 Adios.